So first we'll be looking at the chromosome structure. So the chromosome is basically a one very long condensed DNA molecule associated with proteins and present in eukaryotic cells. So these proteins are large positively charged proteins, and these are the DNA is basically positively charged, and these are attached to these other proteins called histone basically make the whole DNA molecule neutral in nature. So their role is to basically, the histone protein's role is to basically organize this DNA and tightly wrap it around itself. Because we all know that the DNA, the nucleus, is just 6 micrometers in length, in width, and the whole DNA that contains in the nucleus is 1.8 meters. That means that there should be something which should make it inside it should be compact enough to fit inside 6 micrometers, right? So these histone proteins helps in that purpose. So generally we have this chromosome structure which has like two strands. So these two strands are made up of chromatids, and then the chromatid is further basically chromatin, which is tightly coiled around histone proteins. So if we talk about it from big to small, if I were to write it, chromosome, then we will have a chromatid. So basically 2 chromatids will make up 1 chromosome, and this chromatid is made up of chromatin, which is tightly coiled around your histone proteins. And histone proteins have now these chromatin is made up of DNA strands, right? So the DNA, and then DNA contains genes. So if you were to write it in order, that would be the order from big to small. Gene is basically your base sequence of DNA, so whatever like the bases are basically called genes. Now the DNA replicates in the S phase of the cell cycle to create 2 identical strands, and these are called chromatids. So there are 2 identical strands, and these are joined in the center like this, and this is your centromere. We have a structure right here, centromere. So this is joined in the center, and the centromere doesn't contain any genetic material, like there is no genes present, and even the ends of these chromatids have nothing present. The regions are called telomeres, and telomeres basically do not code for any DNA. It's just this extra protection to the chromatid so that when DNA replication happens, there is loss of gene. So to make sure that there is no useful gene which is lost, the telomeres protect the gene from the DNA from that. So there's like 5 stages of your whole cell cycle, which every cell goes through. One is nuclear division by mitosis. We will learn its details later in the chapter. Cytokinesis, this also comes with alongside your mitotic cell cycle. Then there's this 3 stages of interphase, which is G1 phase, S phase, and G2 phase. In G1 phase, basically, it's the gap phase, and there's nothing really important happening here. However, in the S phase, it's basically the cell's resting phase, right? So in the S phase, the DNA replication happens, and also the centrosomes replicate in this stage as well for your mitotic cell cycle. In the G2 phase, the DNA correction happens. So if there's like any error in the in the DNA replication, that part would be covered in your G2 phase, and like everything, like the corrections necessary, would be done in this phase. Then there would be mitosis. Now, what is the importance of mitosis? Mitosis is basically 2 genetically identical daughter nuclei being formed from one parent nucleus, right? So that means that it's also like very important because we have 4 purposes for it. So we have growth of multicellular organisms. So organisms like us, we have like multiple cells in our body functioning for different performing different functions. So we need to have more in quantity, and to like make new cells inside our bodies, we need that. And in unicellular zygotes, it has to grow in. It also helps unicellular zygotes to grow into multicellular organisms as well. So growth may occur across the whole body of the organisms or be confined to certain regions such as meristems of plants. So the roots being grown, or there's cell elongation, that also includes mitosis. Then we have replacement of cell and repair of tissues. Now the best example would be your skin cells, as we know that your skin, the cells on your skin like die really quickly, and also the lining of your gut wears off really quickly as well. So to replace those continuously and rapidly, mitosis needs to take place. Since mitosis is like also a very fast process, so this helps in that as well. Then we have asexual reproduction. This is basically when one parent organism is involved to produce in new individuals, and these individuals are genetically identical. So asexual reproduction happens in unicellular organisms as well as in plants. There are plants which reproduce asexually as well. We have now the cell cycle. We will discuss this in detail. Now, the length of the cell cycle is variable depending on each organism and also the environmental conditions, because we all know that conditions can change and then enzymes working at different temperatures and everything. So all of these affect. So for example, onion root tip cells divide once every 20 hours roughly, and then human intestine cells divide every 10 hours. So there's like a difference between each cell cycle. So interphase includes, during interphase, the cell increases in mass and size and carries out its normal cellular functions as well. An interphase, as I said, contains three parts: G1 phase, S phase, and G2 phase. So at some point in G1 phase, a signal is received telling the cell to divide as it's in its resting phase, and it's just, doubling its mass. That's it. That's all it's doing at that point. Then the DNA in the nucleus replicates and two identical sister chromatids are formed. This phase of the interphase stage of the cell cycle is called the S phase, and S stands for synthesis of DNA. So you can learn it as like that as well. The gap between the previous cell division and the S phase is called the G1 phase, right? The cells will make RNA, enzymes, and other proteins required for growth during the G1 phase. This means that it's increasing in size. Between S phase and the next cell division event, that is the G2 phase, occurs. And during the G2 phase, as I told you, cell continues to grow and the new DNA that has been synthesized is checked for any errors and repaired if there is any. So other preparations for cell division are also made. So like production of tubulin for like the spindle fibers to form during mitosis, that also happens. Then we have nuclear division, which is your mitosis, and this follows your interphase. This is represented as M phase, M in this cycle that is shown. And the cell stops growing at this point. It has like reached its full like size as well, like an adult cell size, and it would stop growing. And then after mitosis, and when two genetically identical daughter cells are formed, there comes cytokinesis, which is basically nuclei are formed in mitosis. So to produce like two genetically identical cells, the cytokinesis will happen, and in cytokinesis, the two daughter cells form, and like the new cell membrane, and like it divides. So in animal cell cytokinesis like constricts the cytoplasm and forms two cells. And in a plant cell, the new cell wall is formed, which like separates the cell then after. Make sure to learn like all the details, because it's very, very, very important to know details for each step. Now, the significance of telomeres. The ends of chromatids in chromosomes are sealed with protective structures called telomeres. Now, these don't have any, this only has like non-coding DNA. It's like, it doesn't code for anything. It's just there to protect the rest of the DNA molecule. And in telomeres, one strand is rich in base guanine and the other strand is rich in complementary base cytosine. Now, this point, you don't need to learn this, it's just extra information. And the main function, as I told you, is just to protect the ends. As you know, after every cycle, the telomere will shorten. Instead of the DNA molecule being the useful genes being affected, the telomere will just, cut down in size. So after a cell cycles, the cell would eventually die, right? So, but there are some cells, such as stem cells, which will, which don't, which don't run out of telomeres. How do they do that? They have this enzyme called telomerase right here. So telomerase is present, and this enzyme basically tops up their like this DNA molecule. So any cell which is like stem cells or cancer cells basically have this, so that they never die out. And if like the cells dying basically is for us is like the aging process as well, so it contributes to that as well. You can see there they have shown the base sequences for your telomeres, and it is like condensed form of guanine. Then we have stem cells. Okay, so stem cells are basically very important. They differentiate into different kinds of cells using mitosis. So what happens is that a stem cell can divide into unlimited number of times, and each new cell produced has the potential to remain either a stem cell or become a specialized cell. So the ability to differentiate into a stem cell again or become a specialized cell, that is called its potency. And there are three types of potency. One is totipotency, pluripotency, and multipotency. Totipotency refers to basically the stem cells can divide or differentiate into any type of cell found in an embryo, or it can differentiate into a specialized cell. It's totally up to that cell. Like if it's required to become a specialized cell, it will become a specialized cell. Then pluripotency cells are embryonic stem cells that can differentiate into any type of cell found in an embryo, but are not able to differentiate into, once again, extra embryonic cells or placenta cells. Then multipotency refers to the multipotent stem cells that are adult stem cells and usually only found in the adult body. And these stem cells have lost like some of its potency associated with embryonic stem cells. So they can no longer be pluripotent. They can only differentiate into specialized cells if needed. We'll just look into multipotent stem cells in detail. And these are, as tissues, organs, and organ systems develop, cells become more and more specialized. So having differentiated and specialized to fulfill particular roles, as we know that we are multicellular organisms, so we need different types of cells to help us function as a whole. So, however, small number of stem cells remain to produce new cells for essential processes of growth, cell replacement, tissue repair. So, although these stem cells can divide an unlimited number of times, because they are stem cells once again, but their ability to produce a different range of cell types is restricted, which is why they're called multipotent. So, for example, the stem cells found in your bone marrow are multipotent adult cells. Differentiate into blood cells which include red blood cells, monocytes, neutrophils, lymphocytes, and all of these. So in adult, stem cells can be found throughout the body. So you have stem cells in your bone marrow, in your skin, your gut, heart, and brain, and they each like divide as required by the body. So research is being carried out on stem cell therapy as well, which is the introduction of adult stem cells into the so different from another individual stem cells are extracted and then put inside another human, which is who needs those stem cells. For example, if a person has leukemia, so the stem cells of a healthy person would be transferred to that person with leukemia, and then hopefully the person would start making red blood cells which are healthy and like not the sickle cell shaped or something. So if you can see here, this is like the zygote and then one sperm cell and then one, what do you call, egg cell combine and then meiosis happens. And then what happens is that these totipotent stem cells make more embryonic cells inside the placenta. And then these divide into pluripotent cells, which can form embryonic and also specialized cells. And then we have multipot, and finally, once like the embryo is formed and the baby is developed, it can have other uh, it will have multipotent adult stem cells, which will differentiate into a limited range of cell types. Then we have how a tumor forms. This is very interesting and a bit confusing as well. But basically, tumors are a ball of cancer cells. We always have confusion between what's a tumor and what's a cancer cell. So yeah, that's that. And then cancers demonstrate how important it is that cell division is precisely controlled, as cancers are caused by uncontrolled mitosis. So cancers start when changes occur in the genes that control cell division. This type of changes is mentioned as mutation. It is called mutation. So the mutated gene, if it's not corrected, then it causes cancer, and this is referred to as an oncogene. So if a cell has this cell controlling cell division gene, it does not possess that, then it will create an oncogene. So mutations are common events that don't lead to cancer most of the time, because in your G2 phase, the DNA nucleotides are the DNA which is synthesized is corrected as well. So most mutations either result in early cell death or result in the cell being destroyed by the body's immune system. As most cells can easily be replaced, these events usually have no harmful effect on the body. But however, if the body is um, unable to destroy these stem cells, then that's going to be problematic. And then what happens is that this type of mutation is harmful for you, and then the tumor starts to form. So one cell, when it divides, if it's a cancer cell, is going to divide and start dividing rapidly and uncontrollably, and there will be no limit to it. So it would just the outside signals would also not affect the cell. It would not cause the cell to die. Then there are agents which also cause your cells to become cancerous as well. So these are known as your carcinogens, and carcinogens include UV light, tar and tobacco smoke, and X-rays. All of these can cause cancer. And if the agent causes cancer, it is described as carcinogenic. Then there are some tumors which do not spread from the original site. So if it's on your, if it's a wart, it would just stay in place and it won't affect any other part of your body or even that part of the body as well. So these are known as benign tumors and they do not cause cancer. So they're just there and it can be removed easily, so they're not harmful as such. But there are some tumors which can move around your body or like spread as well and invade and destroy other tissues. So these are known as malignant tumors and can cause cancer. So these are the dangerous ones. So malignant tumors interfere with normal functioning of the organ or tissue in which they have started to grow, as we will learn later on as well. So malignant tumor cells can break off the tumor from its original site and travel through your via blood or lymphatic system towards other parts of the body and form secondary growths in other parts of the body. The spreading of the cancer in this way is known as metastasis. And the metastasis is dangerous for you as it's difficult to detect in which part exactly the secondary cancer is going to form or has formed. So for this, I have this is a very confusing one. I have one saved right here. This one. Okay, so carcinogens such as UV light, tar and tobacco smoke, asbestos, we don't need to learn this, X-rays as well, will cause your cells to become cancerous. And these oncogenes transformed by carcinogens. So these are called oncogenes now. And then these cancerous cells do not respond to signals from other cells and continues to divide. So it just starts dividing uncontrollably, and other cells will be signaling it to stop dividing or whatever, but it won't happen. Then it will divide through mitosis, and then after mitosis, cancerous cells are not removed by the immune system. The immune system won't be able to remove these. And these just cells start dividing rapidly. And once there's this cluster or like a ball of tumor cancer cells formed, this is called your tumor, and this just gets bigger, and the cells change their characteristic and look different under the microscope. So if like a person suspected of having a tumor, it would be easily distinguishable between a normal cell and a cancer cell. Then the tumor is supplied with blood and lymph vessels, as we know that it's dividing uncontrollably. So it would require more nutrients. So it just takes away the blood vessel and lymphatic vessels surrounding the healthy human cells or like the organism cells, and use those blood vessels and lymphatic vessels to supply itself with nutrients. So tumors, and then the part of it would just break off and also like move inside this red blood cells or the lymphatic vessels. And this process is called metastasis. And the tumor cells invade other tissues and secondary cancers start to form. So the cancer cell would move towards either the lung and cause lung cancer as well. It can also do that. So that is your formation of how a tumor is formed, basically. We just need to learn these steps, and there's nothing more in detail that you would be required in the exam as well. Now comes the main part, which is mitosis. Now, mitosis is basically the process of nuclear division by which two genetically identical daughter nuclei are formed, as I told you, and these are divided into four main stages, which is your prophase, your metaphase, anaphase, and telophase. You can also learn it as PMAT for short, easier to learn as well. And then most organisms contain many chromosomes in the nuclei of their cells, but the diagrams below will show only the general structure. In the different colors, this is like description. So first we have prophase. Prophase is divided into early and late prophase. I have a diagram for this myself. I'll just pull that. 1 second. Right here. So we have early prophase first, which is the first stage in your mitotic cell cycle, mitosis. And in this, you can see that the chromosomes start to appear as chromatin coils up. So if you have a stained cell and you try to look at it under a microscope, definitely an electron microscope, you're going to see these structures very easily under the microscope, because these are thick, dense structures as compared to your single strand, the DNA strands, which are very thin. So this is present, and then the DNA just starts to coil up in the early prophase. And there are two centromeres present, which will start to move as well. So next comes is the late prophase. In the late prophase, the nuclear envelope starts to disappear and break down. And then the centrosomes just start to move towards the opposite poles of the cell. You will say opposite poles and not just up or down or left or right, because that would be considered wrong. You just have to say opposite poles. The nucleolus disappears, and the chromosomes are seen to consist of two identical chromatids. Each chromatid contains one DNA molecule. So these will appear in a proper shape now. And these are attached by your centromeres in the center as well. Then comes your metaphase. In the metaphase, basically what happens is that the chromosomes align on the equator or the middle part of the cell. The proper term is the equator, and this happens when the spindle attaches to the centromeres present on your chromosomes, The centromeres attach to this part called kinetochores. Now, this term is not really required, but it's good for concept and also included in the textbook as well. So, kinetochores are basically these hook-like structures to which these mitotic spindle fibers are attached to, and later they are used to pull the chromosome apart. So the chromosomes will just line up on the equator during metaphase, and each centrosome must have reached the opposite poles of the cell as well. Then comes your anaphase. In anaphase, the chromatids move towards the opposite poles, and these are done by centromeres first, so they will appear in V shape. And when you're drawing these cells, mostly they ask you to draw the anaphase or the metaphase. So during anaphase, you can't draw your what do you call this, chromatid in like straight lines like this, pulling towards your, what do you call it, centrosomes. You will have to make it like a V shape such that it looks like it's being pulled apart towards your, I'm so bad at this, is pulled apart by your spindle fibers. Once this is done, then comes your telophase, which is the last phase of your mitotic cycle. In this, what happens is that the chromatids have reached the opposite poles of the cell, so there's like one pair from each on each end, and then the nuclear envelope starts to reform as well. The nucleolus reforms, and the spindle fiber breaks down because it's not required anymore. Then the centrosome is present on each end, so one will go in one cell and the other will go in the other. And then cytokinesis also happens side by side alongside this. So it basically pinches off in the middle right here, and then two cells will be formed. Cell surface membrane also just expands and divides as well. So this is your mitotic cell cycle. We went through all the stages over there. Then we have this observing, but I'll just go through it briefly. We can't observe live mitosis happening live, definitely, but we can, like, looking at your diagram, you have to draw this in paper 3. They can ask you to draw a plant diagram. So basically what you do is you cut up a very thin piece of either a garlic or an onion, as there are root tips present, so we basically can easily observe these under a microscope, and then remove the tips of the roots about 1 centimeter and place them in a suitable stain to, like, which is basically deep purple or something, and then the stained root tip is gently squashed under your glass slide using a blunt instrument, or just just pressing it with the needle that's present on your microscope. That's your mounting needle. And then you have the cells undergoing mitosis. Images are given below. I'm going to show you different images can be seen, and you can draw them easily. And the annotations can be made to these drawings to show different stages of mitosis. Now this is very digitalized, so it looks very clear. And we can see that this cell right here is mentioned as P, which means it's in prophase. You can see this dense blob in the center. That means that it's in prophase. Then we have this cell. You can say that it's in anaphase because there's like a separation in between, and then there's two, you can see chromatids on each end. What else can we see here? Okay, over here we can see metaphase, this one marked as metaphase, and you can see that the chromatids, or like there's this dense purple color, which is the stain given, and these are the chromosomes, so these will be aligned at the equator right in the middle of the cell. Do we have telophase shown? No, we don't. So I'll show you other images that I saved. These are also from the textbook once again. The textbook has really good images. These are from electron micrograph of cells under a microscope. Okay, so this shows your prophase. So you can see there's like dense structures over here present in the centers. and the DNA has just started to coil up. So you can't see a lot, but you can say that this is your early prophase stage instead of just prophase. Then in picture B, you can see that there's this dense structure right in the middle. This, you can clearly say that it's late prophase, as you can see the chromosomes all coiled up and like in a bundle form. Then we have metaphase. You can see this thin line between the cell. This shows that the chromosomes are lying on the equator of the cell, and these thin fiber-like structures coming out, and they are attached to these chromosomes aligned in the center or the equator. So this shows that the cell is in metaphase. You should be able to recognize the cell micrographs because a lot of questions come from here. Then you can say this is early anaphase that is just about to just separated, so there's like less space, but yeah, they have started to go towards opposite poles. Then the content. Okay, so then this is proper anaphase. This is your proper anaphase. The what do you call the chromatids have reached their opposite poles and they're separate now. Then we have picture F, which is your telophase and cell division, or you could say cytokinesis. What happens here is that the spindle fiber starts to break down, and you can see this nuclear membrane forming, which will just pinch off and divide the cell into two genetically identical cells. Easy and simple enough. A few more things that I would show is this. These types of questions do come in the exam. It's like a very similar type of picture as from the exam. This is taken from the textbook. Now over here, now this cell right here shows you anaphase. As you can see, they're like the chromatids are in V shape. You can see them first of all, and then they're in V shape, and also they're at the opposite ends of the cells. In this is just like in this you can say that it's just in your prophase. This is your this is your cytokinesis happening here. As you can see, there's like they have started to coil up on two ends, and the cell membrane has started to form as well. The cell wall is forming in this because these are plant cells. One more thing that I will show would be this. This type of question usually comes in your MCQs, but also can come in your theory exams as well. And it's basically asking you to deduce the step or like the stage of the cell cycle using the mass of DNA per nucleus. So as you can see, this graph right here shows on the y-axis mass of DNA per nucleus over here on y-axis, and then on x-axis, time is given, and then the graph is divided into A, B, C, and D, and then finally cytokinesis is mentioned. So there are like different masses labeled as the time progresses, and it has asked us that state the phases of interphase shown by A and B. So in A is just like constant, so we can say that the cell is in resting phase, so there's like not really anything happening, so the cell would be in G1 phase. And then in we're saying that the mass of DNA per nucleus remains constant because only the RNA is being synthesized and the enzymes are being created, so there is no, you could say, proper increase in your DNA, the mass of the DNA. Then in B you can see that the mass actually doubles, so like 0.08 was its original mass, and then it increases to 0.06, which means it's double. So double means that the DNA has replicated because it's doubling in size, so two chromatids are formed. So this means that the cell is in S phase over here. Then in C, it has again a constant mass, which means that it's again in a resting phase And this would be after, since it's after your S phase, that would mean it's in your G2 phase. Because in this, all the DNA correction is being happening and nothing else. Then in your in part D of the graph, you can see that it halves almost exactly and back to 0.08, which means that your this is your mitosis happening right here, as the cell is divided into two equal genetically identical daughter nuclei. And then cytokinesis happens and then two new cells are formed. So the mass remains the same throughout. And that's your chapter 5. If you guys have any questions, you can ask.